First of all, I want to acknowledge the contribution of my colleagues, Maria Rodriguez and Lorena Castillejos from our research group, and to Gonzalo Fernández Turren from the University of La República in Uruguay. In this presentation, I will deal with the current status and the future perspective of the use of plant bioactive compounds in dairy and beef cattle. In the first part of the presentation, I will have a short introduction, and then I will analyze the results in vivo that affect rumen fermentation and dairy and beef performance using meta-analysis procedures. In the second part of the presentation, I will address post-ruminal effects that may affect intestinal ecosystem, the immunity, or the metabolic or systemic responses. Plant extracts come from the secondary metabolism of plants. This secondary metabolism, most of them coming from the glucose and pentose metabolism, results in a series of products that are generally classified as saponins, flavones, tannins, and a series of products that are considered generally essential oils. Because of the limitation of time and our experience in research, most of the results that I will show today are related to essential oils. These products are recognized as safe and are used commonly in animal nutrition currently. These products, they have very many different mechanisms of action. However, in most cases, what determines their activity we're looking for in the rumen are related to the interaction with the cell membrane. These products will get into the bilayer of the cell membrane of bacteria and will disrupt the equilibrium between the inside of the cell and the outside of the cell. Bacteria will have to use energy to maintain that equilibrium and will reduce energy available for growth and reproduction. In the dynamic nature of the rumen, reducing growth and reproduction results in a reduction in the size of this specific population. So we have two consequences. Some specific bacterial groups reduce their size in the rumen, while the niche left by this group of bacteria is used by other groups of bacteria to grow and occupy that space. So while some group of bacteria decrease, others will increase. The result of this change in the equilibrium of different populations in the rumen results in most cases in a reduced deamination and peptide lysis, a reduction in the production of methane or inhibition of methanogenesis, and a reduction in the acetate and acetate to propionate ratio, and as a consequence, an increase in propionate and butyrate. This fermentation profile is consistent with a better feed utilization. I will not go in details on which ones and what doses and what are the mechanisms of action in vitro, because there have been published several reviews that address this topic specifically. I will use, however, one review paper that collected 84 different treatments with products and dosages that determined that in most cases what we see is that more than 60% of the cases there are no changes in ammonia, total BFA or propionic acid production. Only in a relatively fair amount of cases there is a reduction in ammonia nitrogen concentration, and this reduction in ammonia nitrogen concentration is consistent with better nitrogen utilization. In contrast, experiments that have shown an improvement in total BFAs or even less in propionate production, they're very small and below 10 and 5% respectively. And in many cases, we actually see a depression on total BFA and a depression in propionic acid production. The interpretation of these studies, and that should be kept in mind, is that most essential oils are not positive to the intentions of changing ruminal fermentation to improve feed efficiency, and we have to look for which ones at what dosages those will be effective. There are several problems with the use of essential oils. The first one is lack of specificity. Monensin, which is often used as a positive control, affects gram-positive bacteria by interacting in the bilayer membrane of bacteria, and there they will make unstable relationships between the inside and the outside of the cell. Essential oils have a similar activity interacting with the biliary membrane. However, in gram-negative bacteria, the situation is different. Gram-negative bacteria are characterized by having an external cell wall with some pores to allow the transfer and circulation of nutrients. Large molecules, like the ones like monensin, are too large to go through the pores and interact with the bilayer. So monensin is not active against gram-negative bacteria. And this difference between gram-positive and gram-negative provides monensin some degree of specificity. This is not the case with essential oils that are in many cases small enough to go through those pores and interact with the cell membrane. As I said, more than being an advantage, it becomes a disadvantage because the specificity is more difficult to achieve. The second problem is the small window of dosages between a positive effect and a negative effect. This is an experiment conducted in continuous culture where you can see that monensin was able to maintain the fermentation, very active, and have a strong effect on the acetate to propionate ratio. However, when you use thymol at lower doses, you're able to maintain fermentation, but not very efficient in reducing the acetate propionate ratio. When you go to higher doses, both of thymol and eugenol, you have a change in the acetate to propionate ratio that is effective and in the direction of the desired changes in rumen fermentation. However, we also have a very negative effect on total BFA production, and that's negative for the utilization of feeds in animals. Finding the right window of activity is not only difficult, but it also imposes risks when applied in vivo conditions. The third problem is that there might be synergies but also antagonisms. And this is an important issue because most of the products today in the market represent a combination of more than 3 or even up to 7 different products at the same time. And to my knowledge, there is no evidence that either synergies or antagonisms between those products have been proven. This is a simplex centroid design, is an statistical design or experimental design where you can mix 3 types of plant extracts, and then you can use this design to draw lines where the type of response is put together with a line. The interpretation is relatively simple. For total BFAs, we're looking for a high concentration. So it's very obvious that the best combination will be associated with large amounts of X2 and small amount of X1. And any time that you increase the contribution of X3, the reaction is negative. It's also true that you could use small amounts of X2 and reach to relatively similar conditions to the combination of these 2 plant extracts. This experimental design not only helps to establish synergies in these sites and antagonism in these sites, but it also allows you to select the right combination of products to achieve maximum total BFAs. For the acetate to propionate ratio, you can use the same criteria to define that in this area is where the acetate to propionate ratio is smaller, and you can play with the different proportions to achieve the objective you're looking for. And again, adding X3 will always result in a reduction on the acetate to propionate ratio. So it demonstrates the presence of synergies and also antagonisms. And the final problem is the doses in vivo. Typically, our in vitro studies show that the effectiveness ranges from 50 to 500 milligrams per liter. If you want to try to achieve this concentration in the rumen, considering a volume of 100 liters and a 10% per hour liquid dilution rate, you should feed about 15 to 150 grams per cow per day of the active component, and this is not realistic. Not only is not realistic, but most effects observed in vivo in research, the doses, the active doses, have been between 1 to 2 grams per day, much lower than this original one. And then if you want to go into commercial settings, then the products contain often amounts below 100 milligrams per cow per day of the active ingredient, which will be equivalent to a much lower concentration in the rumen. Now, why is that happening, this wide range of doses that are decreasing progressively? I have to be honest, and I don't have a clear answer for that. Maybe some of the effects are not related directly to the rumen, and the future perspective that I will talk later are at least part of the answer to this question. So after going into the current status, I'll go to the meta-analysis of the rumen effects and production performance. For these meta-analyses, we have collected only papers from peer-reviewed journals, no abstracts, no reports. Second thing, we have separated both dairy and beef. And for performance analysis, we have only used the studies designed for performance, so no Latin squares were included. We have to consider that this meta-analysis approach has serious limitations in the interpretation, because we have mixed all type of essential oils at all the dosages that have been used in those studies. So it is very difficult to have a clear answer for a specific product at a specific dose, and the reason is that we do not have sufficient experiments addressed with a specific group of ingredients. The meta-analysis in the rumen effects for dairy cow shows that there's no effect in total fatty acids, no effect on acetate, no effect on propionate, no effect on the acetate to propionate ratio, and no effects on ammonia nitrogen concentration. The lack of effect might be due to the dosages that have been used, or they might be the result of the dynamic nature of the rumen, where absorption of acetate, propionate, and ammonia occurs through the rumen wall, and a large proportion of these products also will flow to the lower digestive tract. So those flows may be changing the proportion of these metabolites and hide the potential impact. If we do the same analysis in beef, however, we do see a clear effect on acetate, an increase in propionate, a decrease in the acetate to propionate ratio, and a decrease in ammonia nitrogen concentration. Quantitative differences are relatively small, but they were all significant. The reason why these effects show in beef and were not shown in dairy may be related to the environmental conditions of the rumen. Our previous research showed that those essential oils tend to be more active at lower pH, and they tend to have a stronger effect because most of the effect requires the protonated molecule, and at lower pH the probability of a protonated molecule increases exponentially. This might be the reason why we see those effects in beef but not in dairy. And finally, I go to the dairy and beef performance. There are not many studies conducted for performance, but probably more than I expected before starting this analysis. This is the meta-analysis, the forest plot of a meta-analysis of 28 comparisons. In this meta-analysis, you see the control in the left and the essential oils in the right. And you can see the impact of all the experiments conducted. Some of them had small negative effects, but many of them had actually positive effects. The overall analysis of the meta-analysis results in an improvement of 1.34 liters per animal per day, which is a sizable response. When analyzing data individually, I was concerned about the presence of this type of research. This is one single experiment conducted in Israel that had altogether about 40% contribution to the overall average. Now, these experiments, they fed 4% of the diet as a pomegranate extract, which actually in active components resulted in about 1 gram per animal per day. Now, I just played with the data, deleting that specific experiment because of its high contribution, and the production performance changed from 1.34 to 0.73, about half the amount of the response, yet still a sizable response. If we look at the specific data, we can see an improvement of 4.2% in meal production, a reduction of about 2% in meal fat content, and an improvement in protein content. All these three affect significance. And there was no change in feed efficiency. As you can see, the number of studies included in the meta-analysis has a reasonable size to reach the conclusion that those products are changing performance of animals. If we do the same approach with the beef, we also see a more clear response with a 3% improvement in dry matter intake, a 7% improvement in average daily gain, and an improvement in the gain to feed ratio. All these effects were significant and came out from 29 to 37 different comparisons in these published results. Therefore, we can conclude in the current status summary that essential oils affect rumen microbial fermentation in vivo, and these effects are more clear in beef rather than in dairy. Feeding essential oils result in improved performance in dairy and beef. The optimal dose in vivo needs to be clarified because it's not obvious at what levels those should be fed. And we need to take advantage of potential synergies. These potential synergies may allow us to reduce the amount of essential oils required and may also reduce the risk of having negative effects in this small window of positive results. The second part of my presentation will deal with post-ruminal effects. And this is related to intestinal ecosystem, the immunity responses, and metabolic and systemic responses. Modifying the intestinal ecosystem has been target of research in monogastrics. A lot of this research has been looking at finding a balance between what we call positive intestinal bacteria like lactobacilli versus those that have negative impact like coliforms or Clostridia. A lot of this research conducted in swine and poultry has not been conducted in ruminants. And the reason is that the intestinal microflora in ruminants is buffered by the protection environment of the rumen. So any external potential aggression to this intestinal microbial population will have to survive the rumen ecosystem, which is it's difficult. In addition to that, we don't have major intestinal pathologies in adult cattle, things that do happen in monogastrics. The exception might be that in recent years we've been hearing about the leaky gut, and maybe the leaky gut is a problem that exists, that is limiting production, and where essential oils or plant extract can play a role. Overall, we do have very limited research on the intestinal ecosystem of the production ruminant, and if something has to be explored, I think that it's important to start with a detailed description of the current conditions of the intestinal ecosystem both in the dairy and in beef cattle. Here I'm going to put one example of the potential we have ahead. This is research conducted in swine and deals with the intestinal ecosystem. This is a control versus an oregano oil supplemented pig, and we see the change in the villi morphology. You can see, like with oregano oil, you have much longer and well-defined villi, well-defined crypts, and a much healthier epithelium. This was supported by an increase in the expression of genes related to intestinal epithelium integrity. And you can see that these two molecules that are important in the tight junctions between the enterocytes are stimulated in their expression in the presence of oregano. Therefore, oregano or other essential oils might participate in maintaining the tight junctions that will keep the gut healthier. These, as I indicated previously, may be relevant for maintaining or avoiding the leaky gut. And finally, there's with oregano also a reduction in the immune response at intestinal level, and we can see a reduction of interleukin-1 beta, interleukin-6, TNF-alpha, and MCP-1. All these were strongly reduced in the presence of oregano. So we have opportunities that have been observed in monogastrics that need to be translated into ruminants. Research on the impact of plant extracts on immunity in dairy cattle is very limited. In these studies conducted at the University of Pennsylvania, there was a change in some specific population of lymphocytes, and those lymphocytes are associated with being anti-inflammatory. Now, these effects were obtained feeding 2 grams of curcumin, garlic, or capsicum, and this is a very high amount compared to what we would expect in commercial settings. Moreover, these products were infused post-ruminally and therefore had a direct effect on the small intestine. When doses were lower, about 1 gram of capsicum in this case, there was only reported a small increase in some neutrophils and decrease in lymphocytes, a response that probably was smaller than the previous case. And when the dose was down to 250 milligrams of capsicum, then the only difference observed was a change in the corpuscular hemoglobin. It seems that the response is only observed when concentration of the products are relatively high. Moreover, in these studies there was a long table with very different indicators of immunity, and I have to say that only very few of them actually change. In other studies where the number of lymphocytes changed and as well as the aptoglobin, the doses of jejunal were above 800 milligrams per animal per day. And TNF-alpha increased, but there were between 20 to 30 grams of T saponins. In this other case, 30 grams of T saponins. Again, it seems that something happens, but those effects would only happen if the dose fed to those animals is fairly high. There are also some reports on the antioxidant effects, and these antioxidant effects are, most of them, coming from either phenolics or flavonoids. These antioxidants can have positive local effects on the small intestine, but they also have the potential to affect the transition process in the dairy cow, liver metabolism, and fat metabolism. And it's been important in some of the published papers in helping the transition from prepartum to postpartum, and some suggestions that might also be helpful in the stress conditions between the end of lactation and the dry period, the drying off process. In some cases, the improve in the performance in transition using flavonoids has been substantial. In a couple of studies, the effects have been very clear after feeding in the transition process these flavonoids, with a clear increase about 2.5 liters per animal per day in that peak lactation process, and then also in the overall lactation. Therefore, antioxidants and flavonoids in particular may also play a role in the transition process. We were interested in addressing several studies that have been conducted in vivo with the objective of using these antioxidants to try to improve the quality of the carcass and the quality of the meat. And as you can see, there are between 3 to 7 studies that have used some of these antioxidants in the period prior to slaughterhouse, and they show a reduction in the pH of the carcass, and that was significant. They did show a reduction in lipid oxidation in day 1 and day 7. However, these effects disappear on day 14 after slaughter. And there was also reduction in the cooking losses on day 1, but this effect disappeared in day 7 and 14. As one of the main objectives of this study was changing in color to maintain a better view of this meat after slaughter, and that was not affected neither at day 1, 7 or 14. So there might be potential to explore this area a little further, but the effects appear to be at least not long-lasting enough to be applicable in field conditions. So in summary, for the immunity, we have seen that monogastric research shows different effects that are based on the inflammatory status of the animal and the dose of the product. The studies in cattle are very few, and the effects that we have observed are minor. And in general, when we observe effects, the doses are very high compared to those that are potentially usable in field conditions. Finally, I'll go into the metabolic or systemic responses. Several years ago, a paper published in Nature with the title "The Gut as a Sensory Organ" established the settings for a potential interaction that we should explore. In that paper, they show that the gut has a lot of sensors, taste centers, that sense the presence of fats, sugars, peptones, and biles. These sensors will interact with the specific sensory cells and will transfer either nervous signals to the brain or to other areas of the gastrointestinal tract, and they might also release some messengers that will activate other organs, for example the pancreas, releasing or inhibiting the release of insulin. The interesting thing of these factors is that some of them are directly related to the integrity of the cell, of the lining of the enterocytes, and they are also interrelated with the brain and the hormonal system. And there are sensors in the small intestine that will react to the presence of some plant extracts. So if we learn which sensors we have, what are the effects, and at what dosages will have a response, then we might be able to change metabolism of the animal. And in that direction, the group of Penn State University developed a series of studies with capsicum, and that will serve as an example of the potential. In this glucose test, they observed that animals fed capsicum had a lower tolerance test. The insulin was lower at a challenge with glucose. That means that somehow capsicum was affecting the sensitivity of the body in a general terms, or the sensitivity of releasing insulin. In follow-up studies, they show that hormone-sensitive lipase, and this is the lipase that will mobilize fat from the adipose tissue in the presence of capsicum, was increased. And because it was increased, it means that under capsicum conditions, there is an increase in the mobilization of fat. And other studies show that there was more insulin, but this higher insulin was not able to control glucose, which actually means that there is an insensitivity of insulin. And sensitivity to insulin results in higher glucose, a situation that might be similar to a diabetogenic environment. But more than that, the amount of total non-esterified fatty acids was also increased. Now, in general, an increase in insulin in normal conditions does not allow mobilization of fat, because we have higher insulin and higher fat mobilization suggests that the adipose tissue was insensitive to insulin. This lack of sensitivity can be used to promote mobilization of energy in favor of the energy provision to the animal, and the increase in the increase in glucose in spite of the higher insulin may also help divert this glucose for milk production. The interesting thing is that I've collected a series of studies that they have used monensin as a positive control with 2.3% increase in production performance. This is in liters of milk. A 3% increase with a combination of essential oils, in this case capsicum, cinnamaldehyde and eugenol. The interesting thing is that in any of these that you either include capsicum or use capsicum alone, and in these studies was a rumen protected capsicum, milk production increased much more than what we have been observing in those products that have been targeting the rumen. So it seems that targeting the rumen, we should expect an improvement of around 3%. But with capsicum, there appears to be an additional effect that cannot be attributed to the rumen, because in these cases, capsicum was protected from rumen action, that is actually resulting in a much higher production performance. The last thing that needs to be resolved is that if we are targeting post-ruminal objectives, we have to have an idea of the recovery or the survival of these molecules after going through the rumen. And I have found only a thesis in Austria that reported the data of the recovery of different molecules, plant extracts, after 24 hours in vitro incubation. And most of them are degraded up to 50 to 60%. There are many of them that completely disappear, so they are sensitive to microbial activity, and others that are relatively low. Therefore, it is important that if post-rumen is a target, it's to a very large extent necessary that those products are protected from rumen fermentation. In conclusion, for the current status, rumen effects in vivo are not always obvious like in dairy, but they do work in beef cattle. The performance is improved both in dairy and beef cattle. And the doses in vitro are much higher than those in research in vivo, and those in vivo research doses are much higher than the ones that we normally see in field conditions. And that generates the question whether at the end, in the commercial settings, the effects are related to a rumen effects, or do we really have some post-ruminal effects that need to be explored? As the future perspectives, I think that there's limited research in the post-ruminal effects, and this is a challenge that we have to address. On the other hand, we have enough reasons in animal models and in monogastrics to believe that there is potential to modify the ecosystem in the intestine, the immunity, and the metabolism of cows to enhance production performance. And thank you very much. I'll be open for questions.